Well, it's always wonderful to be in Stockholm. And thank you all for coming to hear about something a little bit different. Clearly, what I'm going to be talking about today comes out of the realization of the last several decades that RNA molecules inside now modern cells, I'm going to be talking about, do a lot more than just play their classical roles in protein synthesis. That is, they are not just messenger RNAs, tRNAs, and ribosomal RNAs, but they do lots and lots of other things. And it turns out that that's true not only of cellular RNAs, RNAs made by cells, including our own, but also of RNAs made by viruses. And so I'm going to be emphasizing viral non-coding RNAs today. And the thing to remember about viruses is that viruses don't make anything up. They acquire things from their host cells. But just because a virus makes something that looks very much like a host piece of machinery that you think you know about, doesn't mean that the virus uses it in the same way. Viruses are very creative, so they have things that look like what the host makes, but they may do completely different things with them. And that's the fun of studying viruses. So I'd like to start with this, what I consider a very profound slide that comes from Australian bioinformaticist John Maddock. And what he has done here is to plot over here the amount of non-coding RNA in an organism. In other words, the amount of DNA that doesn't code for protein directly versus what we might call developmental complexity, which is, of course, a sort of airy term. But what you notice is down here, with 15% or less non-coding RNA, are all the prokaryotes. All the bacteria are down here. When we get to single-celled eukaryotes, they begin to have more and more non-coding RNA. The organisms in here are invertebrates and plants with still more non-coding DNA. And when we finally get up into the vertebrates, and here we are at the very top, with 98.5% non-coding DNA. The other realization that we've come to, this of course has come out of all the sequencing of all the various genomes, but the other realization we've come to in the last 5 to 10 years is that although this DNA is non-coding, that almost all of it is made into RNA molecules inside cells, particularly inside cells like ours. And although these RNA molecules may be very short-lived and only exist for a particular tiny half-life, they could still be contributing to this idea of complexity by doing regulatory things, by doing signaling things. And the question is, what are they doing? Now we know again from the work of the last 10, 20, 30 years that non-coding RNAs do a lot in terms of gene regulation. And here we're talking about vertebrate or mammalian cells. So out in the cytoplasm, we have messenger RNA with the help of tRNA using ribosomes to make proteins. But in the nucleus, we have a lot of non-coding RNAs. Some of the most famous of these are the small nuclear RNAs that are found in the spliceosome. They use base pairing to recognize the ends of introns and form this big machine that then removes the introns from the pre-messenger RNA. We also have some RNAs, non-coding RNAs, that are involved in translation, in this case an elongation of translation. We have some large non-coding RNAs that are involved in altering chromatin structure and turning on and off whole big segments and collections of genes. And one of the most famous ones here is EXIST, which is responsible for turning off randomly one of the X chromosomes in mammalian female cells. we have in the nucleolus small nucleolar RNAs, SNO RNAs, small RNAs that guide the specific modification of ribosomal RNAs at particular places and make ribosomes work better. In germ cells, we have piRNAs, one of whose functions is to protect against the transposition of elements that could occur in germ cells and mess everything up very badly. There are two that are missing. I did not make this slide. One has already been mentioned here. That is the RNP. That's the telomerase that has an RNA template and a non-coding RNA that's used to template the ends of chromosomes. Also, RNase P is not in this picture. It makes the 5' ends of tRNAs, and there's evidence accumulating that's doing more and more in mammalian cells than just that one function. And out in the cytoplasm, we have microRNAs. So these are this tiny class of tiny little RNAs, only 22 nucleotides long, that assemble together with specific proteins and then recognize sequences by partial complementarity in the 3' untranslated regions of messenger RNAs and regulate the protein output and also the stability of these. And there are probably as many as 1,000 different kinds of microRNAs in our cells working in combination on probably 50% of our genes. So they're important for development, for differentiation, certainly for disease, and as all of you know, are a very sort of hot topic of study these days. So these are the non-coding RNAs we know about. But I suspect that these are really just the tip of the iceberg, and we're going to learn about many more different kinds as we go forward. So let me switch, and this slide is meant to overwhelm you, to talking about viral non-coding RNAs. And this is a table we put together a little over a year ago as a review for a book on RNA. And what it's meant to communicate to you is that lots of different kinds of viruses make non-coding RNAs, that these RNAs can be made by both RNA polymerase II and polymerase III in cells. They can be of various lengths, ranging from the size of microRNAs on up to kilobases long. They can be not very abundant or very abundant in cells. Remember that ribosomes in mammalian cells are about 10 to the 7th per cell. So here we have an adenovirus, a non-coding RNA that's 10 to the 8th copies per cell. And many of these others are in the same order of magnitude as ribosomes. So there are lots of them, and they must be doing important things. They all associate with proteins. Some of these proteins are proteins that we know about from cellular non-coding RNAs or RNPs. And viruses also make microRNAs that weren't in the last slide. And again, it's the herpes viruses that encode more microRNAs than other types of viruses. And I think this is probably just because herpes viruses have the biggest genomes. They have double-stranded DNA genomes that encode more than 100 genes. And they probably have space for encoding non-coding RNAs to help them do what they want to do inside infected cells. The non-coding RNAs that my lab has been working on for many, many years, several decades, come from these herpes viruses, from a class called gamma herpes viruses, which are, in fact, primate cells, us and monkeys. Some of these viruses you will know about. Epstein-Barr virus causes mononucleosis. It's also responsible for tumors in B cells. Kaposi's sarcoma virus I'll talk about more later. This is a monkey virus that I'll introduce you to in just a moment. So again, all these viruses have double-stranded DNA genomes. They're oncogenic. They infect lymphocyte cells, so either T cells or B cells. And another thing about them is that they all have both lytic states, where they make more virus, and latent states, where the genome disappears into the host cell and just sits there for extended periods of time before it decides to come out for reasons we don't understand and replicate. The non-coding RNAs that are listed over here, of course, associate with proteins, just like host non-coding RNAs do. And what you will notice from the dates of discovery of these and all the question marks over here as to what they're doing is that these are hard problems. These are the most challenging problems that my lab has worked on, trying to figure out what these viral non-coding RNAs are doing. Are they helping the viral life cycle? Are they helping the virus defend against the evasion tactics that the host will mount against it? We don't know, but that's what we're trying to find out. And recently we've made some progress. That's what I want to tell you about today in terms of molecular understanding. We know more about these particles and understand what at least one of them does. That's led us to some interesting observations about microRNA biogenesis pathways, deviant pathways used by a virus. And I'll tell you a bit about that. And then I want to tell you about this larger non-coding RNA made by KSHV, where we've learned a bit about what it does, but more about how on a molecular level it accumulates to such high levels inside infected cells that it can do what it does. So let me start with the story of a viral snurp that regulates the activity of a host microRNA by selective degradation. And this is the work primarily of an Argentinian postdoc in my lab, Davian Casala, who's had help from a technician. And what I love about this story is that you can think of it as non-coding RNA warfare, because what the virus has done is to make a non-coding RNA that feeds into an important non-coding RNA pathway of the host, to the advantage of the virus. So this comes from herpes virus summeri, which many of you have probably never heard of, and has to do with non-coding RNAs that are called CERs. So it's one of these gamma herpes viruses, and in its normal host, which is a squirrel monkey, it causes sort of innocuous infections. But if you put it into New World monkeys, it causes very aggressive T-cell leukemias and lymphomas. What we work with in the lab are marmoset T cells that have been transformed by the virus. And the remarkable thing about those cells, so these are latently infected cells, is that the most abundant RNAs that are made by the virus are not messenger RNAs for any of the viral genes, but rather these 7 non-coding RNAs called herpes summeri uRNAs. Early on, the virologists that worked on the virus tried deleting one or more of these from the genome, asking, is there a phenotype? They weren't able to pin anything down, but what's quite clear is if you look at these latently transformed cells, if they don't have all of these non-coding RNAs, they grow more slowly. So it has something to do with the growth rate of the transformed cells. Now, the next slide shows you why these are called uRNAs. They look exactly like the snRNAs that are in the splicing snurps. They have 5' ends that are trimethyl capped. They have what are called SM binding sites that bind a heteroheptamer of proteins that are conserved between them and the spliceosome. They have 3' end double stem loop structures, and 3 of them have sequences that are very AU-rich. We focused on these for a long time because there's a particular class of messenger RNAs in our cells that degrades very, very rapidly and is very tightly controlled in its turn on and turn off, called AREs, AU-rich elements. And we thought that maybe the virus might bind up the proteins that regulate that class of messenger RNAs. And when finally we were able, 7 or 8 years ago, to do microarray analyses and look at the host message population in cells that were transformed by the virus carrying the wild-type genome versus we looked at ones that were lacking just SIRS1 and 2 because these are the most abundant and highly conserved, we found no difference in that population of cellular messages. But what we did find was that when the cells were transformed by the wild-type virus, only 7 or 8 RNAs and their proteins were upregulated. And these proteins fit into what's called the TCR, the T-cell receptor signaling pathway. So we finally had a phenotype for the virus, and this phenotype made sense. Because if a virus is going to go into a cell and stay there for months or years without doing anything, it's going to want to make sure those cells keep burbling along and replicating. So it makes sense for them to upregulate the signaling molecules of the cell. That was great, but we had no molecular explanation for what was going on. And at about that time, Heidi Cook, who had done this work, she was an MD PhD student, she left for her internship and residency, and Damien Casala came to the lab, and the first question he asked was, are these T-cell specific functions upregulated by increasing the transcription of the RNAs for their genes? And you can do this by a nuclear run-on assay. I won't show you the results, but what he found was that no, that was not the case. This seemed to be a post-transcriptional effect. And since microRNAs at that point were all the rage, everybody was thinking about microRNAs and post-transcriptional regulation, Damien started thinking about microRNAs. So this slide then introduces you to what you need to know about what microRNAs, how they're made, and what they do in order to understand the next bit of my talk. So what's known from many labs is that microRNAs are made as longer precursors in the nucleus. They can be encoded even within the introns of protein-coding genes. There can be several of them. They're acted on in the nucleus by an enzyme called DROSHA and a double-stranded RNA binding protein that takes them down to precursor microRNAs that are about 70, 75 nucleotides long, the same size as tRNAs. They then go out of the nucleus via a special export system, and in the cytoplasm, the microRNA biogenesis pathway and RNA interference meet because there's this enzyme called dicer that can take either perfectly double-stranded RNA or these imperfect structures down to these 22 nucleotide bits. And they then become associated with proteins, members of the AGO Argonaut family. AGOs are core components of this. And as you know, in the RNA interference pathway, if the complementarity is perfect, then the target gets cleaved. If it's imperfect, one sees either translational regulation or downregulation of the message. And in that way, the microRNAs exert their post-transcriptional control. So what Damien did at that point was simply to look at the sequences of these SIRs, these non-coding RNAs made by the virus. And that wasn't too difficult because there are only about 10 strains of the virus, and he could do this more or less by inspection, and compare it to all the microRNA sequences of the host cell and ask, are there any potential interactions? And so what we're looking at here, the bold red ones are very highly conserved between these strains of the virus. The lowercase, less bold red ones are moderately conserved. The black ones aren't that conserved. And what he was able to find was 3 different host microRNAs that exhibit very good complementarity to absolutely conserved sequences in these viral snurp RNAs. And I should show you now, I should go back to the picture of how these secondary structures look. And what you see is that what's involved from the microRNAs are what are called the seed sequences, the sequences at the 5' ends of the microRNAs, which are most important for target recognition. And they're the sequences that can base pair be complementary to the viral snRNAs. So the question then, of course, was, do these actually interact? Both the SIRs and the microRNAs are wrapped up with their own proteins. Can they interact nonetheless by base pairing in this way? And then what happens as a result? And I'm going to show you one slide next of data but I'm just going to summarize the conclusions, which is that what the viral SNRPs are able to bind, those 3 microRNAs or microRNPs, and one of them, miR-27, which is bound by SIR1, is targeted for destruction as a result of this interaction. And I won't show you all the beautiful data. I'm going to show you just one slide of data. So what we're looking at here is the amount of various different microRNAs. These are all encoded by the host cell in cells transformed either by the wild-type virus that has SIRS1 and 2 or by a mutant virus that doesn't. And you notice that 2 of these, which are isoforms of this microRNA called miR-27, are upregulated in cells transformed by the mutant virus. And we could right away tell it probably wasn't transcriptional because this one, and this microRNA are all in the same transcript. So it had to be something that was happening post-transcriptionally. And what Damien eventually figured out is that inside cells transformed by the wild-type virus that have the partner SIR1, miR-27 very quickly decays. Whereas when SIR1 isn't there, it's much more stable. I indicated that SIR2 binds miR-16, doesn't seem to make any difference. We don't know why this is going on or what effect it has. But quite clearly, in this case, this particular microRNA is having its levels lowered by having the virus in latent form. And it turns out when you trace it through to protein targets of the microRNA, FOXO1 is a transcription factor that's involved in cell cycle and involved in tumorigenesis. And its level then is higher in cells transfected by the wild type that has the lower levels of this microRNA. If the level of the microRNA goes up, then the level of that protein goes down. So it all fits with at least one target. So all the data I didn't show you said that SIRS1 and 2 have these conserved sequences complementary to microRNAs. Damien very nicely showed that they can associate. He did mutational studies showing that it's actually Watson-Crick base pairing and that you can substitute that and change the specificity. I showed you the evidence that SIR1 regulates the level of this host microRNA by inducing decay. And not only FOXO1, the one I showed you, but another transcription factor that's also implicated in malignancies is upregulated. And interestingly, Amy Buck's lab in Britain has shown that for another related herpes virus, that it also downregulates this particular microRNA. The mechanism isn't known, but clearly this microRNA is of interest to this class of viruses. And I didn't talk about this, but the mechanism of degradation is not what our first guess was, that it was also by this ARE pathway. So what I wanted to mention next is that Damien then was interested in what's the mechanism of decay. And several people had reported that seeing microRNAs with short additional sequences on their 5' and 3' ends, thinking that these might be intermediates in the decay pathway. And the way to look at that is simply to do deep sequencing of all the microRNAs inside these latently infected cells. And he did that. He didn't discover anything about the decay. But what he did discover was something very interesting about the biogenesis. If you deep sequence all the microRNAs, you're of course looking not only at the ones the host makes, but any that the virus might make. It was not known that the virus made any, but in fact it does. It makes 6 of them. And they're encoded right in the same region as these uRNA type viral RNAs, and there they are. Let's look a little bit more directly at what's going on. So if you look at the sequences themselves, what you see is that here are the snRNAs I've been talking about. Where they end is where these arrows are. These are the regions encoding the microRNAs that are shaded over, and this is the beginning of the upstream one in each case. And what's in between is something called a 3' box, or a 3' end processing signal that several labs have investigated over the years that's involved in maturing the ends of snRNAs. And this juxtaposition was so very close that it made us, of course, wonder, could it be that in this case the microRNAs are co-transcribed with the snRNAs? So with a new postdoc in the lab, Ming Zi, they simply laid down primers here and here and did RT-PCR and showed that, yes, indeed, there seems to be one common precursor. And that, of course, then raises the question, what's the processing pathway? Is it different from what's used by most? And here I just review again what I showed you earlier, that the first processing is by Drusha DGCR8, but usually in that case, the microRNA exists in a imperfectly double-stranded region, but that extends farther than the end of the microRNA, and I told you about dicer. What's known about snRNA processing is that a big complex that involves a protein called integrator 11 recognizes the 3' box and cleaves upstream. I seem to be running out. Is there another? Do you still have your beautiful blue one? That would be great. Int 11 is a protein that's homologous to the CPS73, which makes the cleavage to which poly-A's are added at the ends of messages. So this is a endonuclease. And then what happens to snRNAs is they're bound by a protein that's a disease gene in spinal muscular atrophy, one of the most common genetic causes of infant death, and that assists the SM proteins getting on. So the question was, how does this chimeric precursor get processed? And again, I'm not going to show you any data, but just tell you that Damien and Ming have taken two approaches. One approach is to mutate the signals that we know are recognized and ask what happens. The other approach is to knock down the proteins in the cells and ask, what do you get? And let me just summarize what's been found is that, of course, the special promoter is necessary for the appearance of both the snRNA and the microRNAs. The SM site, which stabilizes the snRNA, is necessary for that, but doesn't affect the processing of the microRNAs. And the 3' box that would cleave apart this part and that part is absolutely essential for getting both of them. And indeed, what Damien and Ming found is if you knock down Drosha DGCR8, they're not required. Dicer is required, and the integrator complex is required. So we have a novel pathway for processing microRNAs that proceeds through first cleaving here and then generating the snRNA, and then in steps that we need to find out more about for making the mature microRNAs. So before I summarize this, I want to go on and say that this is not the first time that herpes viruses have been recognized as making microRNAs from unconventional precursors. It turns out that MHV68, a murine herpes virus, makes a tRNA-like molecule that downstream has microRNAs. And this comes from work from Brian Cullen's lab and Linda Van Dyke's lab, two different examples. And their biogenesis requires cleavage by RNA-Z, which cleaves at the 3' end of tRNAs in order to separate them. So viruses do very, very interesting things. So I'd just like to summarize here and then go on to the last thing I want to talk about. So we need to work out this co-processing. We still need to work out the degradation mechanism. We need to find additional targets of this microRNA and figure out why this microRNA is somehow antiviral and viruses like to get rid of it. We need to figure out why some of the snurps bind microRNAs, and we don't know what they're doing. But the really interesting questions are there cellular counterparts of these snRNAs that also regulate the endogenous microRNA population? And are there host microRNAs that are co-transcribed with snRNAs or other non-coding RNAs upstream that then use this unusual novel processing pathway? And we already have hints that that may be going on. So the last thing that I want to talk about then is a completely different virus and a completely different RNA. So this is one made by KSHV, Kaposi sarcoma herpesvirus. It's called PAN RNA. You'll see why in a moment. And here the focus is on an element, an RNA element in the RNA that's very important for preventing the degradation of that RNA in the nucleus so it can get to these very high levels and do what it does for the virus. And this work started with Nick Conrad, who's a postdoc. He now has his own lab at UT Southwestern, Vula Mealy, who is also a postdoc who's in Virginia, MD, PhD student, undergraduate who's now at Stanford, etc. And I want to start out just by reminding you, KSHV is this virus that affects people that are already immunocompromised, causes these horrid lesions in people with AIDS. And what we're going to be talking about is in its lytic phase, when it's making more virus, it makes something called PAN RNA for polyadenylated nuclear RNA. I'll show you that in a moment. And this RNA looks like a messenger RNA. It has a cap transcribed by Pol2. It has a long poly-A tail. But it doesn't code for anything. It doesn't have any introns. It never gets out to the cytoplasm. It's never translated into protein. It accumulates to extremely high levels inside of the nucleus of the late infected cell and accounts for as much as 80% of the polyadenylated RNA inside the cell. So you take the total messenger RNA, most of it's this particular RNA with its poly-A tail. So what's shown here is a situation late in infection. All this PAN RNA in the nucleus. There are host messenger RNAs that are being downregulated in a way that I'll mention in just a moment, and the viral RNAs are the ones that are getting successfully translated. So how does this work? We don't have a total answer on that, but what I'm going to focus on is this element called the ENE that's located just upstream of the poly-A site. And what the PAN RNA does when there's so many copies of it in the nucleus. It turns out the virus has a function that helps it shut down the host messages and allow the virus messages to be translated. This isn't really understood because they look just the same. But one of the things that that function does is to move the poly-A binding protein, which is a very important protein in the cytoplasm of all cells for getting messages translated. It moves it to the nucleus, and that's part of the shut-off effect. And when it gets to the nucleus, it binds to all these copies of the PAN RNA because they all have poly-A tails and there are tons of them. And what we've learned by getting rid of the PAN RNA is that if it's not there and does this, then the late viral messages don't get translated and we don't get any infected virus production. We don't know the mechanism. But this slide focuses on this element called the ENE that I do want to tell you a little bit more about. And that's necessary for getting the PAN RNA to these high levels. And its activity, I'll say right now, and you'll see why in a moment, is dependent upon the cutting and the adding of the poly-A tail, which is part of the process of making PAN RNA and making all messenger RNA. So in this slide, I want to show you a very sort of vivid visualization of what the ENE element can do. So it's been known from the earliest days of cloning that often if you remove the introns from a gene you're trying to clone and then put it back into mammalian cells, it just doesn't get expressed very well. You put an intron into it, fine. And you see that this is a beta globin gene that's one of these classics. And what you see here, this is a northern blot, you see this level of beta globin messenger RNA if you have the 2 introns. 1 intron is enough, you still have a very high level, but if you remove both introns, what I just told you, the level drops down to sort of less than 5%. And you can rescue this expression by putting this piece of PAN in that has that E&E element in it in a longer copy, a minimal copy, 1, 2, 3, 5 copies, not in the reverse orientation, which just tells you it's working on the RNA level. And what Nick Conrad and Vulamili figured out is that what the E&E is doing, it's rescuing the RNA, polyadenylated RNA that it's in, from a very rapid decay process that occurs in the nucleus. And so if you don't have introns and you have a poly A tail, you can undergo a very rapid decay that has a half-life of about 15 minutes, whereas with the E&E, the half-life is extended to about 4 hours. Or if you put the intron back in, the RNA is stabilized and exported to the cytoplasm. So that's the other way of solving the problem. But there are many polyadenylated nuclear RNAs that are stable, and the question is, do they have this element? And some of them do at least. So how does the E&E work? I haven't shown you a proposed secondary structure, but Nick's idea, based on many experiments, was that maybe what happens is that this internal U-rich loop in the E&E, in fact, binds the poly A tail and sequesters it. And this is attractive because the first step in decay is often exonucleases chewing on the poly A tail, and once the poly A tail is gone, then other decay processes just take away the molecule. So if you can protect the end, then that will serve to stabilize the molecule. And we had indications from studies of other non-coding RNAs that structures like this might be able to be formed. So the last thing I want to mention to you or tell you about is the work of Rachel Mitten-Fry, a postdoc, who took on this question of what is this interaction, and actually did a high-resolution crystal structure with help from Jameen Wong and Tom Steitz's lab to figure out really what's going on. And the results were not what we expected. So first of all, there are 2 things here. She wanted to make sure that the E&E could interact not just with a poly A tail that's downstream, but one added in trans. So this is just a small oligonucleotide, A10. And she also wanted to take off extra sequences that we didn't think were important. And what this gel shift slide shows is that the affinity for the full-length E&E is the same as for a truncated complex that's missing this stuff up here. So she had something simple to try to do structural work on. She did some NMR, and then she got crystals. And I show you this just because it's such a beautiful picture of a crystal structure. But what it really says, so at 2.5 angstroms resolution, what Rachel saw was not anything like a SNO RNA interacting with a ribosomal RNA, but actually formation of a triple helical structure using that loop. And here I tell you a little bit more about it. So here are 2 views of this triple helical interaction. So it's not at all like this, and I'll show you that again later. So specifically what's going on is that 5 of the A's make Watson-Crick base pairs with the right-hand side of this internal loop. And then Hoogstein base pairs to form a classic UAU triple are made with the U's of the other side of the loop. And then those interactions are extended by 3 what are called A minor interactions with the GC base pairs that are at the top of the stem. And so specifically then, just to remind you, here's the standard Watson-Crick interaction, and here's the Hoogstein with the additional U. So what we're looking at here is UAU, 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 etc. This stack of 5 is the longest stack of triple helical interactions that's so far been observed in any naturally occurring RNA molecule. And here is an overlay of those triples. They're so perfect that you almost don't see that it is an overlay. And what I find particularly wonderful about it is that this is exactly the structure that was predicted by Gary Felsenfeld, David Davies, and Alex Rich in the 1950s when they studied strands that were made of 2 poly-U's and 1 poly-A. That's just exactly what they predicted. So this just compares Rachel's structure. There are shorter bits of triple helix, like three triples in some other structures that have been done. It's not at all like the snow RNA interaction. This is NMR structures from Peter Moore's lab and from Julie Feigen's lab, not at all like that. And just to say a word about the A minor interactions, they are very much more common and have been seen all over the place in ribosome structures. And we have an example of the 3 different types of them going deeper and deeper into the minor groove, and that then stabilizes the overall scene. So Rachel, being an excellent scientist, said, oh, this is very nice that I have this triple strand, but is it really biologically important? Is it necessary to stabilize the poly-A tail? And what she noticed was that in Nick's earlier work, he had mutated the central U's in that internal loop, changed them to C's, and found that these mutations debilitated the ability of the E&E to stabilize. What she then realized is that a UAU triple is isosteric with a CGC triple. And she wanted to know whether if she put a single G residue into a long poly-A tail, would this structure find that G and would it then rescue the phenotype? And here's the experiment. It's done in nuclear extract with a sort of synthetic substrate that has a 60 long A tail that's been added. And what you see is that in the extract that some of the molecule survives intact after 60 minutes, but not if you delete the E&E or if you make these substitutions. On the other hand, if you put a single G residue in either very close to the end or more in the middle of the A-rich stretch, the E&E is able to scan along through the sea of A residues, find that single G clump in, and stabilize very, very specific lengths, just as you would predict, either a longer length or a more moderate length of the poly-A tail. So that's very, very good evidence then that this interaction through the triple helix that is extended by the A minor interaction is biologically important for the function of this element in stabilizing. And of course, as I said earlier, that raises the question, where did the virus get this? Are there cellular RNAs that have E&E-like elements that do the same thing? And bioinformatically, we're onto some of them. We're currently testing them, and we will definitely see. I wanted to end with just a little bit more sort of evolution. When we started working on PAN, virologists told me, they said, KSHV is really weird. It's a human virus. It's the only virus that makes this PAN RNA. But human viruses sometimes are really strange. Just because it's in KSHV doesn't mean that you should expect to find it in other herpes viruses. But other herpes viruses do have shut-off effects. They do have relocalization. And once we knew that you can substitute the UAU with CGC, we could bioinformatically search a little bit more intelligently, and Kaziu-Takowski has in fact found E&E-like elements in rhesus radenovirus and in, this is equine herpes virus. And so here you see the very same sorts of things. This one has been tested, It actually is part of an RNA that's very much like the PAN RNA. And here on these 3 different genomes, you see the clustering of other proteins in the vicinity, so it's clearly syntenic in location. So at least several other viruses have a PAN RNA, probably the same sort of function. We're clearly looking harder at all these others because what was going on was that nothing was conserved but the structure. And once you know how to look for the structure, then you can find the homologs. Okay, so the organizers said I should end with a question that I would like our research to answer. And I think this is pretty obvious from what I've been saying all along, is that viruses have provided us with incredible insights into things that host cells do. And I confess to having great fun with these viruses, and the question is, how many more things about non-coding RNAs will viruses be able to tell us? And so here is Rachel Mitten-Fry, who did the crystallography on the ENE, Damien Kazala, who discovered the snurp that is responsible for degrading a host microRNA, and Ming Zi, who's working on this new novel pathway of microRNA biogenesis. And thank you all for being here and listening.